Voilà, là, il y a 10 participants qui viennent d'arriver.
You are in the meeting room. Recording in progress.
Bonjour à tous. Nous sommes en train de régler les derniers détails pour mettre en route le séminaire. We are now working on the detail to have the webinar soon. Test micro. Test micro. C'est bon. OK, it's working. Tu peux juste vérifier que tu entends bien tout sur le Zoom.
Le problème, c'est qu'il va y avoir un écho. Parce qu'il y a des gens qui vont parler dans le webinaire.
Non, ils écoutent juste.
Ils écoutent juste. Ouais. Dans ce cas-là, tu peux parler dans le micro et on coupe le son.
Non, il faut que tu parles dans ce micro-là, c'est important.
Dans ce micro-là, on va rester.
Voilà, tu peux sur la télécommande là.
Ça sert à quoi, ça?
Tu as le bouton du volume. Quand tu as fini, tu peux l'éteindre. Vous en avez jusqu'à quelle heure pour laisser
1 heure.
À 1 heure de l'après-midi
Non, 12h30. On en a pour 1 heure.
Ah, ok. Je repasserai alors.
Ouais. Ok. Ok.
C'est clair.
Je te laisse. Okay, so we can start this seminar. Thank you for coming in the room, and thank you to people on the web that follow this webinar. It's always a challenge to do a hybrid seminar. I hope you appreciate behind your screen, but I encourage every people in Montpellier to come in the Thaler room. It's always better to be in person, but don't worry, we will stay hybrid during all the year. So if you cannot come, the webinar will work, hopefully. We are just beginning. So I let the introduction now of this seminar.
Good morning, everyone. So I'm very happy here to present Maryse Vanderplanck. So Maryse just joined the CNRS last year. She got permanent position. She works at the CEFE. She has defended her PhD in 2013, where she, in the University of Mons in Belgium, she worked during a PhD on the impact of generalist metabolites on the ecology of wild bees. Then she started a postdoc where she's been interested in the impact of specialized metabolites on the ecology of wild bees in collaboration with Neuchâtel in Switzerland. Then she has been interested in the effect of climate change and climate change on the ecology of bees and plant interaction. And she just started some project on the nutritional resilience in bees and plant interaction. So thank you, Maryse, for being here.
So thank you for this introduction. I hope the microphone is working, I guess. Okay. So thank you, all of you, for coming or connecting to listen my presentation. So thank you also to the organizing committee for inviting me and give me the opportunity to share with you some of my research. So as said Magali, I'm specialized in the chemical ecology of bee-plant interaction, and more specifically, I am studying the chemical composition of pollen and its role in shaping the bee-plant's interaction. And today I will share with you a little journey through some of my research and tell you about the nutritional resilience in bumblebees. But before going deeper in this quite a specific topic, I would like just to remind you of some generalities about the bees. So I'm pretty sure you all know the honeybee. So it's this one that form a quite big swarm at the end of the spring. And it's a species that have been semi-domesticated by the humans in hives, for instance, for the honey production. But actually, the honeybee is only one species among the 20,000 bee species that have been described around the world. And all these bee species present a really high diversity in their ecological and functional traits. So in their shape, in their color, in their size, their behavior, their phenology, but also their physiology. But despite all these differences, they also have some common points. And the main one is that they all share a long and intimate evolutionary history with the flowering plants. because the plants rely partly on the bees for the pollination and the reproduction, and the bees in turn rely exclusively on the plants for their nutrition and then their reproduction as well. So as I said, the floral resource is the exclusive food resource for the bees. So they use mainly nectar at the adult stage because they need a lot of energy for the flight and the foraging activities. And the nectar is really rich in carbohydrates, so a source of energy. But they mainly just feed on pollen at the larval stage because they need a lot of proteins for the grow, but they also need lipids for the development. For example, they use specific sterols as precursor of the hormone for the molt. So bees have to interact with the plants just for their survival and for their life cycle, but they can do it in different ways. And so they can have different levels of floral specialization. And I would like to draw your attention on this notion of floral specialization because it refers specifically to the pollen collection and not to the nectar collection, because the bees can forage nectar on a larger range of host plants than they do for the pollen. And so there is a lot of different adjectives that allow for describing these different levels of interaction between the plants and the bees, so these different levels of specialization. But here I have chosen just to present you the simple distinction between generalist and specialist species that is done for herbivores in general. And so for the specialist species, it's the bee species that will forage on a very restricted range of different plant species that all belong to the same plant families, while for the generalist species, the species will be able to forage on many different host plants, many different species that can belong to different host plant families. So you can have up to 15 different plant families. So regarding our bumblebees, they are mostly generalists, so they are able to forage on different host plants. So for example, Bombus terrestris is able to forage on 100 different plant species, and these species can belong to different plant families. But of course, as for everything, there is some exception, like for example, Bombus consobrinus in the northern part of its distribution, and also Bombus gorsteckeri. These two species are tightly associated to the Aconitum genus, so it's a floral genus that is that occurs mainly in the mountainous region, like for example in Switzerland, and that has a really peculiar pollen composition. But being generalist does not mean that the bees will forage randomly on all the available plants, but there is also, like for the specialist species, a selective foraging behavior. Like for example, here for the bumblebees, you can see that there is different host plant integrated in the pollen diets. But for example, the two last plants that I have presented here, that are Asteraceae plants, are never included in the pollen diet and are only foraged for the nectar. So such selective foraging behavior is allowed because the bees are able to discriminate among the host species by the use of different signals that are emitted by the plant that they are able to perceive. So it could be olfactory cues, visual cues, but also chemotactic cues. And there is some evidence that the chemical composition of pollen also play a role in this host discrimination and then in this host selection by the bees and here by the bumblebees. So actually, the pollen is a really complex chemical matrix that has major compounds like the proteins, the lipids, but also the carbohydrates, but also minor compounds like minerals, vitamins, but also some phenolic compounds. And usually, like for the other part of the plants, we can make a distinction between the primary and central metabolites and the secondary and specialized metabolites. So for a quick definition, the central metabolites are all these metabolites that will be directly involved in the development, the survival, and the reproduction of the plants. So you have the proteins, the lipids, the carbohydrates, but also the DNA. While the specialized metabolites will be rather involved in the interaction of the plant with its environment, so with the biotic and abiotic components of its environment, like for example, the attraction of pollinators, the deterrence of the herbivores, but also the protection against UVs, for example. And you have really a lot of different compound families in these specialized metabolites, like for instance, alkaloids or flavonoids. So just to have a look in the variability of central metabolized in pollen, you have here different plant species presented in abscissa and the polypeptide contents, so it's the protein contents in ordinate. And you can see that according to the plants, you have a really different concentration in the pollen. Like for example, the Echium vulgare is really low, while the Medicago and the Odontites are quite higher. And actually, these three species are foraged by the bumblebees. If we move to the variability for the specialized metabolites, here again, you have the plant species in abscissa. So it's all the Aconitum genus, the famous one that you can find in the mountainous regions. And in ordinate, you have the total alkaloids content. So it's a secondary metabolite quite close to the nicotine that you can find in cigarettes. And you can see that here again, even within the same plant genus, you have a high variability with some plants that display pollen with a lower concentration and other with a higher concentration. And all are again foraged by the bumblebees. So you can see here that the bumblebees are really they have to face to this high variability in the chemistry of pollen. And this variability is among plant species, but also within plant species according to the environmental condition. So if the plant is exposed to high temperature, the composition of the pollen will be different if it's a plant that are exposed to optimal temperature condition. And so all this variability and all these chemicals that we can find in pollen actually will have a strong impact on the reproduction of the bees and the bumblebees. So it can impact the reproduction in terms of number and mass of offsprings, but also in terms of the developmental time of the larvae, but also in terms of larval ejection. So the larval ejection, it's a specific behavior of the bumblebees workers. When they are exposed to different stressors, they will start to eject living larvae from the brood. So this is really something you can observe, for example, when they are exposed to some pollen with nutrient deficiency, or if there is a heat wave or something that is really stressing for them, they will just start this larval ejection for the brood. So here you have an example of one of my study. You have in abscissa 3 different pollen diets. So you have the Salix, Cistus, and Cirsium. So the Salix is a highly suitable diet for the bumblebees. The Cistus in the middle is medium suitability, and the Cirsium is a really bad suitability. Actually, it's this Asteraceae species, so it's one that is never foraged for the pollen in nature normally, but only for the nectar. And you have here the larval ejection and the mean larval mass that is produced by the bumblebees. And so what you can see here is that for the worst diets, so this one from the Asteraceae species, you have a higher ejection rate inside the brood. And you have also smaller larvae that are produced compared to the microcolonies that have been fed with the other 2 diets. You can also have an impact on the health status of the bees, so the immune response, cellular response, but also the survival and mortality rate. And so here again, I will give you an example. So in this experiment, the bumblebees have been fed during 5 days on a really high suitable diet, so the Salix one. And after the 5 days, they were either kept on the good diet or switched to other diets, so medium suitability or low suitability. And we have measured a variation in the prophenol oxidase levels. So the prophenol oxidase is an enzyme that is involved in a non-specific immune response of the bees. It's like the melanization process. I don't know if you know about it or not. And so here what we can see is that we don't have any difference regarding the variation in the active phenoloxidase or in the total phenoloxidase among the different diets. But if we have a look within the diets, we can see that between the total and active level, we have a significant difference for the Salix diet, so the highly suitables. And that means that there is a high stock of inactive phenoloxidase, so prophenoloxidase, that's available for the bumblebees. And so it's likely that the bumblebees will be able to mount quite quickly an immune response to face a challenge like parasites or pathogens, for instance. And last but not least, all these parameters will, of course, have an impact on the foraging behavior of the bees, so on the host selection, but also on the resource collection. So here there is some studies. In the first one, I have just shown that the bumblebees avoid in nature the pollen that is not suitable to support the development of their colonies. In the second one, I have shown that for the Asteraceae, there is different chemical and mechanical traits that are not suitable for the bumblebees. They are not able to extract the nutrients, or there is nutrients deficiency or some toxic compounds. That's why it's never quite never used in nature by the bumblebees for the pollen collection. And in the last study, I have just put all this data in a more evolutionary perspective, and I have shown that actually the chemical composition of pollen may have driven some shift from host plant in the evolution of bumblebees. So of course, I am not the only one working on this topic. And so there is a lot of different research teams around the world that work on a similar topic, and they have shown that actually the bumblebees are able to detect and to taste the quality of the pollen. So they are able to discriminate among different protein-lipids ratio, but they are also able to discriminate between different concentration of the same nutrients. So for example, fatty acids or amino acids. And then according to this detection, they will flexibly forage on the different plants and always select the pollen that is the most adapted to their physiological and nutritional requirements. So they are able to taste the pollen and then to choose the good one for their health status. And so this ability of detecting the pollen quality and shift to the pollen that is the most suitable for them is really important in the current context of pollinator decline and global changes. So you all know that there is different environmental factors that has been identified as stressors for the bees and that are responsible for their decline. So for example, you have the changes in the land use that go along with reduction of floral resources. You have also the spread of pathogens and parasites, also the use of pesticides, the global warming, and of course the occurrence of alien invasive species. So it could be other bees, other plants, but also other parasites. And so my main research hypothesis is that if you provide the bees with the good pollen, they will be able to find some chemicals that will allow them to cope with these stressors in the nature and then to decrease the impact of the global change on their physiology. And this is exactly the definition of the nutritional resilience that I will use during this presentation. So of course, we aim to test this hypothesis of nutritional resilience to face different environmental stressors. And the first one we have tested is the changes in the land use and the reduction of floral resources. So you all know that the human have a strong impact on its environment. So you have the urbanization, but also intensification of agricultures that lead to quite poor floral landscapes or really homogeneous floral landscapes. And so you have a modification in the floral resources that are directly reflected in the pollen diet of the bees and here of the bumblebees. So to check for this, we have first performed some palynological analysis for 5 different bee species during 2 different time periods. So we have just analyzed the pollen load, see which species are present for one period and for the second one. And then we have calculated the change in the diet contribution. So it's what it's put here in ordinates. And we have then used also some modelization studies to see how the distribution of the plant species change between these 2 time periods. And so it's what is presented here in abscissa. And you have the full line that present the relation between these 2 parameters for each bumblebee species. So you can see here that for most of them, there is a trend to forage on expanding species, expanding plant species. And this is also significant for the Bombus lapidarius. But you can see that for here, you have quite a different response. And this could be explained by the different ecology of these bees. So the 4 other species are quite generalist and are able to forage on many different plants. But this one, even if it's a generalist species, it has a difference in its tongue. Actually, it's longer than the other species. And so it preferred forage on plants with long corolla tube, like, for example, the clover. And these plants are not expanding. So it explained that here we have a trend that is totally different from the other species. So if we look into details to the change in the pollen diet of the Bombus terrestris, so it's the most common bumblebee species in Europe, you can see that between the two time periods, you have many huge change. You have some species that appear, that increase, some that disappear, some that decrease. And here, the most important thing is that you can see that in the new diet, if I can tell it like this, you have two newly dominant plant species. It's totally different from the other one. So we have decided to test the suitability of these two newly integrated and dominant diets for the bumblebees. And to do so, we have used bioassays using microcolonies. So microcolonies consist of some workers. So usually we used 5 workers that we put in a plastic box. And among these 5 workers, one will become the dominant one and will just behave like the queen. So she will start by laying some eggs and these kind of things. And these microcolonies are provided with syrup to mimic the nectar and with pollen, and we can choose which pollen we want to feed the bumblebees with. And so we do this experiment during 1 month, and we are able to measure different parameters that could be related to the resource collection or to the reproduction or to the stress response with, once again, the larval ejection that we just have a look before. So here you have the different results. So in Abscissa, you have the different plant species. So once again, you have the highly suitable diet, the medium suitable diet, and you have the 2 newly integrated resources that I put here in green. So in yellow, you have the pollen collection, and in blue, you have the pollen efficacy. And so the pollen efficacy is a composite parameter quite often used in this kind of bioassay, and it corresponds to the total mass of offspring that have been produced by the microcolony divided by the total mass of pollen that has been consumed. And so you can see here that there is no difference for the pollen collection, but there is a slight difference between the rubus and the trifolium regarding the pollen efficacy. But anyway, it remains totally included between our 2 controls for medium and highly suitable diets. So that means that even if the bumblebees change their diet according to the expansion of species, they do it in a way to select the plants that remain suitable for the development of their colony. So it's like a kind of plasticity that they are able to detect the quality and then to select the plants they will newly integrate it to be sure that all the physiological requirements will remain met. And so here we can talk about nutritional resilience. So we then decided to move on to another stressor, so the global warming. I know you know about it, but I will anyway tell some word about the context. So the surface mean temperature has dramatically increased and is still predicted to increase. It depends from the scenario. You can see that in some it's really a big increase, in others it's like more controlled. But anyway, besides this increase of the mean temperatures, you have also the increase in frequency and intensity of the extreme events, like for example, the droughts, but also the heat waves. And I am really interested in the heat waves here because it's something that is known to strongly impact the physiology of the bumblebees. Actually, when the bumblebees, the males, are exposed to really long heat waves, it can alter the sperm quality and then really dramatically decrease the reproductive success of the bee species. So once again, to test the possibility for resilience regarding the heat waves, we have used lab experiments with microcolonies, and we have used a totally fully crossed design under control condition. So we have 3 different thermal conditions. We have a control, we have a short stress with a heat wave of 5 days, and a long stress with a heat wave of 10 days. And for all these thermal conditions, we have fed the bees either with a highly suitable diet, a medium suitable diet, or a low suitable diet. So here it's the Taraxacum, but it's an Asteraceae like I presented to you before. And we measure the different parameters I have told you just before this slide. And here is a part of the result we have obtained. So you have in Abscissa the different thermal conditions, so the control, short stress, long stress, and you have 2 different kind of results. So you have the mortality, and in the upper part, the broad mass, and you have a different color code according to the suitability of the diet. So you can see here that yes, the heat waves are really impacting both mortality and reproduction of the bees, but the impact is different according to the diet they are feeding on. So you can see here that, for example, for the low suitability diet, the broad mass really decrease, but it's still quite okay for the highly suitable diet. And also you can see that the mortality increase for the low suitability diet, but it remained quite stable for the high suitability diets. So these results show that if you provide the bees with a highly suitable diet, it can mitigate the negative impact of the heat wave on its reproduction and mortality that you can observe in the microcolonies. So it allows for just keeping a good performance of the microcolonies. And so this can be considered like a nutritional resilience again. We then decided to move on again and have a look to the spread of parasites and pathogens, along with the occurrence of invasive alien species. So regarding the spread of parasites and pathogens, you all know that there is a lot of new emerging bee diseases that are mainly due to the spillover from the managed and invasive pollinators to the wild ones. And you can have different transmission modes. So you can have vertical transmission, so between the hives or between the colonies of the bumblebees. So it's just from the adults to the offspring. But you can also have horizontal transmission modes. So it could be between different bee species, but also within a same bee species between different individuals. And this horizontal transmission mainly occurs during the floral visitation, because the flowers are contaminated by the infected bees, and then the parasite is transmitted to healthy bees. So it's why it's drawn just here. And regarding the invasive alien species, yes, they are more and more present in our landscape, but their effects are quite controversial, because sometimes it could be beneficial because it could facilitate the pollination of other plant species that are present around. Sometimes is neutral, and sometimes is really detrimental because it can totally disrupt the pollination network in a location. So here we have decided to mix the field observation and lab experiments. So we have selected different locations, some that are invaded by the Himalayan balsams and some that are not. And in all these locations, we have sample walkers on another bumblebee species here. So it's Bombus pasquarum. And then in the lab, we have performed a PCR analysis just to try to quantify the presence of Apicystis bombi. So this is a parasite that is shared between the honeybees and the bumblebees. Critidia bombi, which is a specific gut parasite of the bumblebees, and then different Nosema species. So you have Nosema bombi, Nosema serranae, and a cluster of several Nosema species we are not able to distinguish using our PCR protocol. And so here is the results. So you have, again, in abscissa, the different parasite species, and in ordinate the prevalence. You have in pink the invaded species, and in green the uninvaded species. And you can see that here, when the balsam is present, you have a decrease. You have a trend of a decrease for the prevalence of the different parasites, and this is significant for the Apicystis bombi. So what's it important to say here? It's because we have field observation. You can tell, okay, maybe there is some confounding effects that count for this effect, that it's not the balsams that is responsible of this decrease in the parasite prevalence. And we thought about this also. So we have checked for the genetic diversity of the bumblebees that were present on the different location and was exactly the same. And we also just have a check on the flowers, the richness, abundance that were present before the flowering peak of the balsams. And it was also equivalent between the different locations. So that means that the effect that we observed here is really due to the balsam and not to another confounding effect. And so actually here, we can see that having access to this peculiar resource had decreased the parasite prevalence, or at least significantly for some parasite species, for the Bombus pasquarum. And we have made some analysis of the pollen chemistry, and we have found a specific molecule, so the ampelopsin, that is known for antimicrobial effects. And so it's probably these chemicals that is responsible for this effect and this case of nutritional resilience. Then we have decided to really focus on the spread of parasites and pathogens, and we have decided just to try to distinguish two different categories of medicinal effects. So you can have therapeutic effect and prophylactic effects. For the therapeutic effect, is more like to cure an existing infection and then to reduce a parasite load. And for the prophylactic effect is more to prevent an infection to establish and then to result in a loss of the parasite infectivity. So once again, we have used microcolonies to do this research, but this time just the setup was quite different because we just had an initiation phase during 4 days where the bumblebees were only fed with syrup. And then during 15 days, we have fed them with both syrup and pollen. And we have tested, yes, different type of pollen. So you have the heather, the sunflower, the poppy, and we have just used 2 different controls here. So still the one with the highly suitability, so the Salix one, but also a multifloral pollen diet because it's the control that is quite often used is this kind of study about medicinal effect. And we just wanted to have a control to be able to compare to the other extant experiments on this topic. So for the prophylactic treatment, we have inoculated the workers during the pollen feeding phase, so 2 days after the feeding. So that means that the bees had access to the pollen before being infected. And for the therapeutic effect, we have made the inoculation 2 days before the feeding. So it means that the bumblebees was already infected and the infection was efficient before the feeding start. And so, of course, during the experiments, we have just monitored the parasite loads at quite constant time intervals, so the day 4, 7, 10, and 13. And so here is the result. So I'm sorry, because there is like quite an heterogeneity in the data, so everything is like in just the down part of the graph. But you have the number of days after inoculation in abscissa and the parasite load in ordinate. And we use global additive mixed models here to make the analysis of the data. And what we detect is like there is a significant difference with the heather and sunflower pollen that allow for controlling like the parasite load that were lower than for the 3 other pollens. And this is mainly due to the day 10 here. And normally also just before this day 10, it's not so obvious at the end of the experiment, but I can tell after the logarithmic transformation of the data, it's more obvious, but I didn't have time to show it here. So we move on for the prophylactic effect, and here it's more obvious. So we still have the number of days after inoculation and the parasite load. We still use this additive model for statistical analysis. And here you can see that even at the end of the experiment, the heather pollen and the sunflower pollen allow for having a lower parasite load in the bumblebees compared to the other 3 pollen diets. So we can say that here we have a nutritional resilience, and we can either tell about medicinal effect of the pollen. And this study is really important because it shows that you don't have to wait that an environmental stressor is present just in the environment to have an action, because prevention is also important. So before having something, you have to just provide the bees with the good diet. So it's really important to have this kind of measure now and not wait for catastrophic stressors in the environment. And this is really in line with what I have shown before with the prophenol oxidase. I show you that when we fed the bumblebees with the good diet, they have a higher stock of enzyme that can be activated just to be able to mount quickly an immune response. And so here you can see that, yes, if you provide prophylactically, I don't know if it's okay, but you know what I mean, the bees with the good resources, they will be able just to have a better immunocompetence. So all these results are to be taken with caution because I present you different stressors individually, but outside they are all like in the same time. So you can have some synergetic effect that are rules that I have presented here. I also told you about bumblebee species, but it's really like a small drop in the ocean of the bees diversity. And all these results are really context dependent. So that means that what you observe here, maybe it's not exactly the same if you go in America or in Asia. So you have really to take into account like the heterogeneity of the conditions. So in time, but also in space to address this kind of question. And so just for you to know about this importance also of the bee species and this diversity of bee species, here you have a study from Drossard and colleagues about the bee decline, but at the Belgian scale. So you can see that on the more than 300 species that have been evaluated, there is a third that is threatened. So it's really not only the bumblebees. You have a large range of bee species that are concerned by the global decline. And this experiment is really interesting because it's also taking into account the sensitivity of bees according to the functional traits. So you can see that according to the host plant range, the sociality, but also the nesting behavior, you don't have exactly the same response of the bee species. And what is interesting here is like for the host plant range, for example, you can see that the opportunistic bee species with a strong preference are more impacted by the global decline than the other ones. And if you remember, it's exactly the case of the bumblebees that I present you with a long tongue that are really related to the flowers with the long tube corolla. So that means that you can be a generalist, but if you have a strong preference for one plant species, you can be touched by the decline as well. And so all of these studies and all of this last thing I show you just highlight the urgent need for taking into account all this functional diversity of pollinators when you are just talking about resilience, nutritional resilience. So you have to take into account the host plant range, the nesting behavior, the phenology, the sociality, the tongue length as well. And so it's exactly what we would like to do here in the project that we start at the CEFE. So if you are interested in this question of nutritional resilience, stay tuned because it's only the beginning of the story. And we hope we will find the solution to save the bees around the world, of course. So I present you a lot of different studies. And of course, I don't do all these studies on my own. I have help from several colleagues. So they are here, put it on the screen. And I would like specifically thank Natalie and Antoine that helped me with the immune aspect of the bees, but also with all this medicinal effect of pollen, parasite inoculation, because there was like a lot of trials before being able to inoculate the parasite or to measure the prophenol oxidase, all these kind of things. And I would like once again say thank you to the organizer and also thank you to you. And if you have any question, I will be really pleased to try to answer and to discuss with you about this topic. Thank you.
Thank you. Maybe we start with taking questions in the audience. So are there questions?
Hi, thank you very much for your talk. This is just a question about your experimental designs. I see that your sample sizes are pretty small, like 9 or 10. And also in slide 11, if I saw, you said that you looked at 5 different species of bumblebees. And then like you also like had microcolonies for them, right? So I was wondering if you sampled your microcolonies from the same or like the different areas. So I was just wondering from where you picked them, or did you change the individual composition of your microcolonies over time for your experiments?
Okay, thank you for the question. So for the microcolonies is more often the Bombus terrestris that were used because it's a commercial species that are available all the year. It's quite difficult to find the queens in natura, and it's also quite not advised to collect them because it's like the queens is the foundress of the colonies. So if you go and you tend like, for example, 10 queens, like it's a disaster for the place where you are. So it's better to try to use commercial species or to try to have like a rearing in continue in the lab, but it's quite difficult to have like the control for all the bumblebee species because there are some that are pollen storer, other that are pocket maker, for example, and it's different way to forage on the plants or to have the pollen inside the colony as well. So here it's really on the buff-tape bumblebee, so this one with the white butts that I was working on and that was commercially available. And we use different colonies to make the microcolonies to have like genetic variability among the bumblebees. But inside the same microcolonies, you have to put all the bumblebees from a same mother colony because they are fighting together. They are not recognizing, so just they kill each other if they are from other colonies. So we have like, for example, I don't know, 5 different colonies, and from each we do like 3 microcolonies, and we have all these microcolonies in the same treatments. So that is fully crossed for the origin of the bees, but also for the treatment and all these kind of things. And regarding the number of replicates, of course, it's really huge management. You have all the microcolonies of bumblebees. So even like if the replicate is 10, if you have like 5 treatments, you have 15 colonies that you have to just fed, to look for the larval ejection, to be sure that you don't have any mortality, to provide the syrup. So like it's really, really challenging and quite sportive as well. And everything is also done in a controlled condition. So you are in a room that is quite limited in space as well. So you cannot have like an infinite quantity of different microcolonies because it's just impossible. But anyway, when we did this kind of experiment during the statistical analysis, we just know that according to the replicates and to the normality or heterogeneity of data, we choose like the best model to adjust and be sure that the results are reliable enough to be published.
Are there other questions in the room or online?
No question online.
How can a bee know what is good for its offspring? Does it have the same digestive enzymes, or does it have some kind of feedback from its offspring, or how can it work?
Yes, actually there is like the adults itself that can detect it because there is specific receptor on the antennae and the legs and also on the proboscis. Yes, but also on the tongue. It's not proteins. It's only highly hydrosoluble components. So normally it's more about fatty acids and amino acids that can be detected by this kind of receptors. And then you have like a feedback after ingestion for the proteins and lipids, but you also have a feedback from the brood. Like if the larvae are not developing well, they have like different communication through pheromones that indicate to the workers that the pollen is not good, and so they can change. And it has been shown during experiment that if you let the brood inside the colony or you remove it, the workers don't have exactly the same response according to the pollen collection and the choice of the resources. So it's quite sure that you have a broad feedback for the selection as well, but only for the social species, as you don't have it for the solitary one.
I understand how it can work in a, let's say, a social group, for instance, a small colony, if you feed them all with the same pollen. But bumblebees, if I'm right, don't communicate, so they forage alone. So
no,
do they know how
they do communicate as well. They also have the communication. It's not the dance like the honeybees, but they also have a system of communication.
So they will really know what their offspring are eating.
Yes, exactly. You have like a feedback from the offspring that allow the adults to adjust the resource collection and also the selection of the plants.
Thank you. Are there other questions? Online? Are there questions online?
Hi, thank you for the talk. I'm very happy that you joined the CEFE. I just wanted to ask if you know what is the degree of in the variability and strategies of foraging, whether you know like how much plasticity and how much genetic diversification there is. So, for example, due to a population structure or altitude or whatever?
Yeah, actually, I cannot fully answer for the genetic part. For the foraging strategies, it really depends, yes, from the altitude and the condition. Like, for example, I know that when you are close to the limit of the distribution and normally quite far from the optimals in temperature, for example, regarding the species distribution, they normally tend to forage on a more diverse different host plant to be sure they will pick up the good nutrients, for instance, and they are more specialists in the center of the distribution when the conditions are better because they can have a strong preference. They are not so much need because it's less challenging for them. according to the latitude, you have difference also from the altitude, sorry. You also have like some difference, like for example, we know that at higher altitude, as the climatic conditions are quite harder, you can have different behavior, but also like in terms of sociality, like for example, normally, if you are bumblebees from one colony, you cannot go in another one. But in high altitude, it's allowed. Like you can stay for one night, for example, because they know that maybe you don't have time to go back to your side. I don't know to tell it. It's quite anthropocentric, but you have like altruism that is better presented when the conditions are quite hard. But regarding really the plasticity of the foraging behavior, it's still quite unknown, to be honest. So.
For the climate change perspective to know if it's more plastic or
yes, exactly.
I expect that if they like this thing that you said for high altitudes like populations, so I guess that this is genetically determined.
It could, yeah. Yeah, it's really something that could be interesting to investigate, of course.
So actually, maybe I have a question, or maybe two that are related regarding your prophylactic experiment. How did you inoculate the parasite?
So actually, as it's a gut parasite, so it will be quite disgusting. So if you want to go out, I can accept. You use the feces from infected bees, then you purify it, you have the parasites, and you have like to count the different parasite cells. You have a known load, and then you give it, so the feces, in a sugar solution to another bumblebees, and you wait that all the drop is consumed, and then you consider that the bee is inoculated and infected. And 3 days after, you have to check if there is a parasite load in its own feces to be sure that the infection is effective.
Is there parasite growth? Like you didn't really talk about the parasite and how, like if what you saw was due to the growth of the parasite population. So like what's the generation time of this parasite? Like, you know, how does it work? Because the underlying question is, could it be kind of I don't know, like a confounding effect? So it's not prophylactic, but it's just that your diets differ. And maybe for some reason, the metabolism differ between your treatments. And in that case, that could influence the growth rate of the parasite, but kind of indirectly.
Yes, but it's exactly what we would like to test. So even if it's like direct or indirect effect, our main point was to show that there was prophylactic effect anyway. But we know that for some diets, like for example, the either one, like there is a specific compounds that infect so this parasite have a flagell, and it's this flagell that helps them to just stay in the gut of the bumblebees, and that make the infection. And so the flagellum can be lost with some specific metabolites, and so it's directly flushed with the feces, and so the infection is not able just to establish inside the individual site. It's a known effect for the either nectar, for example, and so maybe it's the same for the pollen, we don't know. But here, what we did for this specific parasite only, because it's really different according to the parasite we are investigated. So here for the Critidia, we only count the flagellate form because it's this one that are known to be infectious. So we don't have a look to the other forms because it's quite difficult to recognize the asthmatozygote form, for example, against cells debris or something like this. So we really concentrate on this form. We are sure it's the infective one.
And a related question, it's about I guess you assume that the parasite load is negatively correlated to fitness, but I'm wondering about, you know, that maybe some diet, they increase your parasite load, but they increase your survival of your reproduction. And so in the end, did you test this? Like you know, what do you think is the relationship between what should be your conclusion regarding this prophylactic effect? Like, you know, if you have a bad diet for your parasite load, but a good diet, yes, it could be a good diet for your nutrition, right?
Yes, exactly. So it's exactly the difference between host tolerance and host resistance. So host resistance is just you will have an effect on the parasites, and then the host will have a better resistance because of this effect on the parasites, while the tolerance is that the diet will have like a good impact on the fitness of the individuals that will be able to mount better an immune response, for example. So it's two kind of different effects. You can have the impact directly on the host fitness, and then as the host is better, it can respond better and just fight the parasites.
My question was just about that, you know, the pollen is providing nutrients directly to the insect.
Yes.
So you could also have a direct effect on its survival through protein or through carbohydrates.
This is the host, what is called in immunity. This is called host tolerance.
Yeah, but it's independent of the immunity. Like, for example, females, they would use lipids to produce the eggs, and maybe say that pollen with a high concentration of the lipid is increasing your parasite load, but then the female will produce more offspring because of this. So in the end, they don't care about, you know, the parasite, because even if they get infected, you see what I mean?
Here I only present the parasite, but we have measured all these other parameters, and so we take into consideration the trade-off when we just give all the results. But we don't have any, for this one I have presented here for the parasite load, we don't have any effect, beneficial effect on the diet on the bumblebees that will allow to have a different trade-off. So it's like quite the same regarding the fitness of the bumblebees for all the diets, but it's different for the parasites. So you don't have any trade-off in this study. But yes, of course, it can occur, and we have to have a look to all the parameters. It's only one part I presented here.
Thank you. Are there other questions in the room or online? Okay, so I guess thank you again.
Thank you to you.
Nice talk. See you next week.